Welcome to the Organic Chemistry Podcast, Dr. Brian Lloyd's scribblecast of organic chemistry lectures and solutions to homework problems. In this scribblecast lecture, we're continuing looking at our classes of lipids, and having finished off our look at neutral acyl glycerols, we're now going to press on and look at several other classes, important classes. The first one we're going to look at is class number 2, waxes. Now, waxes are derived from different sources and vary greatly in their chemical composition. The most common wax you're familiar with, paraffin wax or candle wax, is not a true wax. It's composed of a mixture of solid straight-chain hydrocarbons. Carbo wax as well is a synthetic polyether. However, a true wax or naturally occurring wax is a monoester formed from an alcohol and carboxylic acid. In this case, a monoester of a fatty acid, a long chain fatty acid, and a monohydroxy-alcohol. Now, long-chain fatty acids typically are from about C16, 16 carbons, to 36 carbons long, so you can get some very long-chain fatty acids, and a monohydroxy-alcohol. So an alcohol that's straight-chain has one hydroxy group. And these alcohols are also typically very long chain, also from C16 to C36. Now, there are several properties that are very important to waxes. Waxes, as you are probably familiar with, are soft and pliable. When they're warm. And they become hard when cold. They have higher melting points than fats. And they are more resistant to oxidation than fats. All right. The most important biological role? The most important biological role is to serve as protective chemical coating. Protective chemical coating on the surfaces of animals and plants. So they're found on the skin, fur, feathers, leaves, and the cuticle of exoskeleton of insects. Let's take a look at some examples. The first wax we're going to look at is lanolin. Lanolin or wool fat is a mixture of fatty acid esters of the sterol. We're going to see this sterol later on. It's a mixture. The alcohol end is a steroid, so it's called a sterol, and its name is lanosterol. I'll show you the structure of lanosterol when we actually cover steroids. And agnosterol. So we got lanosterol and agnosterol. These combine with fatty acids, various fatty acids, to make lanolin. Okay. Okay. Beeswax. Beeswax is made up of several constituents. It's secreted by bees and used in the making of honeycomb. Myrrhichyl palmitate. Myrrhichyl palmitate, okay, an ester of myrrhichyl alcohol, so it's made up of myrrhichyl alcohol, which is C30, H61, and OH. Okay, that is myrrhichyl alcohol. If you react that with palmitic acid, you get the ester myrrhichyl palmitate. One key constituent, okay, and this guy's palmitic acid. One important constituent of beeswax, myrrhichyl palmitate. So it's an ester of these two. Another important wax is Carnauba wax. Carnauba wax is obtained from the leaves of Brazilian palm. A typical molecule that's labeled, and there's several, so we got a long chain alcohol end here with 34 carbons, and on the other end we had a 22 carbon fatty acid. So you see they're just linear esters with very long chain carbons. It has a high melting point, and because of its hardness, resistance to wear, has been used in floor polishes and car waxes. So there are three examples of waxes. Lanolin, okay, which is connected with wool. Beeswax, which is connected with the honeycomb. And Carnauba wax, which is used in waxes for floors and automobiles. These are naturally occurring waxes, remember, have this ester structure. Other waxes that have similar properties and behave, they're soft and pliable when warm, hard when cold, but don't have the ester functional group are not natural waxes and not true waxes based on fatty acid chemistry. Well, that's all we're going to say about waxes. We're going to now move on to our little more complicated third classification in our classes of lipids. And that third classification is the phosphoacylglycerol. Which is sometimes called a phosphoglyceride or phospholipid. So you can also call it a phosphoglyceride or phospholipids. Now, phospholipids are lipids that contain phosphate ester groups, inorganic ester groups that can contain phosphate esters. Inorganic esters based on phosphoric acid. Essentially, what you have is your standard glycerol molecule reacts with two fatty acids. So we got our standard carboxylic acid. And I'm going to just use an R group attached here. And now these two fatty acids may have the same group. They may both have the R side chain, or one may be R prime, which is a different group. So these could be different. And a phosphoric acid, HOP, double bond O, double bond O, H2PO4, oh sorry, H3PO4, duh, thinking of sulfuric acid. There we go. Okay, when these react, you get a precursor to your phospholipid. You get what is called a phosphatidic acid. So for example, you might have R prime, C double bond O, O, CH. And then you have a CH2, O, C double bond O, R. And you get CH2, we get an O, POH, double bond O, OH. This is called a phosphatidic acid. Now, phosphatidic acid occurs in the L configuration, and L-phosphatidic acid only occurs in very small amounts in cells. But note, it's an important intermediate in the biosynthesis of phosphoacylglycerols. Right? What essentially happens to phosphatidic acid is some kind of alcohol group, and I'm going to put R double prime to indicate it has a different R group. This group attacks that alcohol position, so that what you end up with looks like this. Okay, so we end up with a new group, and these groups, these molecules that make up this HOR double prime alcohol can vary, and we're going to look at the 5 most prevalent ones. Now, one thing I want to say before we actually look at those specific structures is that this molecule can exist in equilibrium. So let's draw it again at the top and consider what it would exist like. And because of the phosphoric acid group, this phospholipid can actually deprotonate because it has an acidic group, so it's in equilibrium. And at a pH of 7, okay, or at more neutral pHs typical in the cell, the actual phosphoric acid group is in its deprotonated state. So this is what you typically get at around a pH approximately 7. This type of structure. So remember that when we actually look at our groups. So what we're going to do is we're going to put certain groups on, and the first group we're going to look at is, and so let's draw the structure. Okay, what is the first group we're going to put on here? Well, the first group, group 1, is based on the molecule ethanolamine. And ethanolamine, when protonated in acidic conditions, appears like this. The amine end can pick up a proton. The ethanol ammonium cation can react, and that would produce this species. Now, at a pH of 7, my OH deprotonates on the phosphoric acid and becomes a minus. So at a pH 7, this molecule is neutral. It's a Zwitter ion. So at a pH 7, this molecule would be a Zwitter ion. The second group, choline, is based on the ethanolamine, the ammonium cation. The only difference is in the molecule, the 3 H's on the nitrogen become 3 CH3 groups. So if you make that change, you have a phosphatidyl ethanolamine or a phosphatidyl choline. And right, I've got here, in naming these, notice the name is based on phosphatidic acid. You drop the ic acid, add YL, and you name the group that's attached. So this is phosphatidyl ethanolamine. And ethanol means one word. Phosphatidyl and ethanolamine are two separate words. So phosphatidyl ethanolamine, or sometimes it's called ethanolamine phosphoglyceride. The common name for phosphatidyl ethanolamine is Cephalin. The common name for the choline derivative is Lecithin. Okay. Lecithin and Cephalin. Okay, notice that whether you're ethanolamine or choline, at a pH of 7, you get this Zwitter ion, the minus on the oxygen off the phosphorus and the positive on the nitrogen. So overall, the molecule is neutral. Now we're going to look at all the different ones first, and then we're going to compare their structures and take a look at some of the interesting structural features common to all these phospholipids. The third group is the amino acid serine. So if I replace this group, group 3 is a serine unit. Now, do you remember serine? Serine was an amino acid, okay? Serine was an amino acid that had this structure, HO, CH2, attached to our basic CH, CO2H, a basic fragment for amino acids. So if we attach this OH on the end, we get a CH2. And remember, amino acids form their own Zwitter ions. So we'll draw amino acids in their standard Zwitter ionic form. And if you use serine, you'll notice you have two negatives now and a positive in the overall phospholipid. In this case, you end up at pH 7, an anion. So the serine example generates at a pH of 7 an anionic molecule. Now the serine is referred to or named as phosphatidylserine. Phosphatidylserine. Okay. Now, what's interesting about this is the functionality of these different phospholipids differs with the changes that you actually put in the phospholipid when you attach these groups to them. Group number 4 is based on our carbohydrates and therefore might qualify as a glycolipid. And that is inositol. Remember, inositols are 1, 2, 3, 4, 5, 6 hexahydroxycyclohexanes. An inositol has alcohol groups off of all carbons of the cyclohexane ring. So here is an example of an inositol. And you can attach such an inositol to the phosphate group. If you do, and if I use the same inositol, I have an OH up, OH up, OH down, OH up, OH down. So here is a phosphatidyl inositol. Phosphatidyl inositol at pH 7 is an anion because the alcoholic side chain doesn't lose any protons. So again, we have a polar head. Lastly, we use glycerol in our backbone. You can actually, the fifth group is just add another glycerol to the molecule and produce a phosphatidylglycerol. Now when you say phosphatidylglycerol, you're not referring to the glycerol backbone that has the two fatty acids and the phosphoric acid, not this part. We're actually talking about, when we say a phosphatidylglycerol, adding a second glycerol molecule to our phosphatidic acid. So a phosphatidylglycerol. So if we do that, and we put a glycerol on here, you end up with phosphatidylglycerol. Again, at pH 7, this would be an anion because the alcoholic group would not lose any protons, and you'd have the negative charge. Okay, what do all of these phospholipids have in common? Ethanolamine, choline, which makes cephalin and lecithin. Serine, phosphatidylserine, inositol makes phosphatidyl-inositol, phosphatidylglycerol. What do they all have in common? Well, if I focus on, let's say, the lecithin molecule, which is based on choline, I have, and let's use some fatty acids here. Here's one CH3, bracket CH14, and C double bond O. We have in all of our phospholipids a hydrophilic head. Overall, this head is neutral, but it does possess ionic groups which water can hydrogen bond to, so it's hydrophilic, meaning water attracting. In this end of the molecule, we have a hydrophobic tail. Long carbon chain that's neutral. And in many ways, this is reminiscent of a structure that we've seen previously. Polar head, nonpolar tail. It reminds us of a soap. Now, phosphoacylglycerols are called amphipathic polar lipids. They're amphipathic in that they have this hydrophilic head and hydrophobic structure. So that's a new term. Amphipathic simply means it contains two, if you like, different polarities in their structure. And it's because of that, they're polar lipids. One end of the molecule being nonpolar, one end of the molecule being polar. Now, you may be familiar with this, except the difference with the soaps is if we use a circle to represent a polar head, now we have two nonpolar tails coming off. And certainly this is the type of thing you've seen in cell wall structures in textbooks. You'd often see these heads aligned thusly. In what is called the phospholipid bilayer, and this would continue. And this phospholipid bilayer is the standard structure found in cell walls. Now, it's thought that the amphipathic structure of phosphoglycerols plays an important role in the structure of cellular membranes. Phosphoacylglycerols may be important in the transfer of proteins across membranes and, as well, activate some enzymes. Phospholipids are abundant in the brain and nervous tissue, egg yolk, seeds of plants. They circulate in the blood loosely bound to protein in the form of lipoproteins. Now, phosphatidylethanolamines and phosphatidylcholines, as I've drawn here, are phosphoglycerides found principally in the brain and nerve cells. They're also found in egg yolks, wheat germ, yeast, and soybeans. The difference in polarity of the head, the size and shape and electric charge of the head, affects the nature of the phospholipid. The phosphate group has a negative charge, as we point out, pH 7. And as we summarized previously, ethanolamines and cholines, are dipolars with their ions. Overall, no net charge is shown here. But at a pH 7, serine and the inositol and glycerols have a negative charge. The variations in size, shape, and polarity all play a key role in the structure of the various types of cell membranes. Okay, well, there you have it. 3 important classes of phospholipids so far we have covered. We're not going to spend much time on the fourth class, which is called the glycolipid, other than to say glycolipids are important, and they represent fat molecules that have carbohydrate units attached to them. Okay. Next Scribblecast lecture, we're going to look at the fifth and very important group referred to as prostaglandins. Prostaglandins are a very biologically important class of fatty acid-based lipid. In fact, prostaglandins are so profound in their biological activity. They're extremely potent and often have been referred to as one of the most important class of lipids ever discovered. And so we're going to take a closer look in our next Scribblecast lecture in the structures of prostaglandins. Well, I'm Dr. Brian Lloyd. This is my Scribblecast lecture. I thank you for listening. Thank you very much.